to this class. So when we talk about milk and milk products, as we discussed previously, there are so many kinds of milk and milk products. The type of microorganisms in each of these products are different, and invariably the final the kinds of microorganisms that are the ultimately the result of either introduction of the microorganisms from the cow itself or the introduction of these agents through the milk supply chain. That's what we are going to discuss. What are the kinds of these microorganisms, what kind of deleterious effects they bring about in the milk and milk products, and if at all their number exceeds certain threshold number, what are the implications on the product shelf life and the public health. So before entering into the microbial aspects of the milk and milk products, let us understand the different mechanisms by which the udder homeostasis is maintained. When we talk about the different sources of microbial contamination of milk and milk products, the first and the foremost contributor to the milk supply chain is the animal itself, the milk animal itself. And when we talk about the milk animal itself, either it is the health of an individual cow that has given the milk, or the health of the bunch of cows, so whose milk has been pooled. That's what is the contributor number one. And second aspect is the udder health of these animals. We know that large number of microorganisms, mostly the pathogens, could be secreted into the milk from a cow. And second thing is that large number of microorganisms could be entering into the udder through the environmental source. These are the things that we have discussed. So udder homeostasis forms an important basis for the total contingent microflora in the milk and milk products. And when we talk about the udder homeostasis, you know what is homeostasis. So the udder defense mechanisms, they act against the microbial colonization and modulate the complex interactions between the microorganisms and the host itself. The factors that determine this homeostasis include the cow genotype. Certain breeds of cow, they are naturally resistant to certain microorganisms. That's the number one. Number two is in this physiological status of the cow. We say mastitis as a disease of high producing animals. So more is the production, more is the risk. And the other characteristics in the conformation of the udder and its susceptibility to injuries. These are the factors that some cows will have saggy or more protruding type of udder that is more prone to the injury whenever animal makes sit-ups and it moves, so that is more prone to the injury, and such thing is likely to add more microbial flora to the milk than a cow whose udder is held high. The second B is the invading microorganisms. Number one character, the A is all these factors related to the cow. B is all those are factors that is associated with the microorganism. Whatever may be the defense mechanism of the cow, whether it is in the intramammary or the health of the cow itself, if a pathogen is very strong enough, is having a lot of virulence factors, a lot of pathogenesis factors, obviously they are more dangerous because they are having infectivity. So they infect the udder, leading to the mastitis, and whenever there is mastitis, the number of microorganisms in the milk is going to increase. And third is in our control, that is the managemental practices. The managemental practices, keeping the cow barn hygienic and clean air, all those things, and udder hygiene, milking hygiene, animal hygiene, the hygiene score. So they are the management related aspects. Indirectly, they will contribute to the milk microflora. As we know that when we talk about the total number of microorganisms in the milk, it is A plus B plus C plus D, like many. Okay, one is A is the cow itself, second is the udder, third is the milker, fourth is the air utensils, then other utensils and the equipment that are used to handle the milk. So these are all the factors associated. So ultimately, what I stress here is healthier is the cow, lesser is the microorganism count in the milk. Okay, diseased is the cow, more is the count. Healthier is the udder, lesser is the count. Infected is the udder, more is the count. Healthier, hygienic are the practices that are followed, lesser is the count. So unhygienic are the practices. Unhygienic is the environment, more is the load. So these are all the factors that influence the total number of microorganisms in the milk, and so also in the milk product. One added factor here is the contamination that occurs in the processing of the milk, leading to a milk product manufacture that counts a lot. So the diversity of the microbiota, okay, other microbiota, microbiome, what we call it as biota, biome, they are alike. When we say it is dependent on a locality, it is dependent on the ecosystem in which the cow lives. So the ecosystem you can understand. So is it an extensive type of husbandry? Is it an intensive type of husbandry? What kind of intramammary infections the cow is more susceptible? So these are all the things that determine. And the important aspect is the prior to calving, what kind of management that has been taken. So in India, it is not a practice to go for the dry cow therapy. So you know that this is the entire milk production cycle of a cow. Okay, so there is a transition period and early lactation, mid lactation, late lactation, and there is a dry period. So after that, there is a calving. Then after calving, again early lactation starts. Like this, the production cycle of a cow, depending on the parity, it is the first and the second, first parity, second parity. Likewise, it's going to continue. Several 7, 8, up to 12 parity will get the cows. And when we talk about how the cow was managed during the dry period, okay, as I told you, when we talk about the Western world and India differs here because we do not, except for some organized dairy farms. So dry period management is not there. So during dry period, if cows are infused with intramammary infusions, so obviously they affect the type of microorganism that is there in the cow udder, and that's going to add to the milk later on. So with antimicrobials, without antimicrobials, the type of the microflora, the microbiota, what we call it as, is different. So in general, when we talk about the microbiome, so it is more of a cow oriented, more of a management oriented, more of a production system oriented than a generalized one. So generalized one, we do have certain generalizations with respect to understanding the microbiome when it comes to the milk, but it is specific to a particular system of production, I would say. So my MO here means microorganisms. Okay, we know that cow body itself is an ecosystem for large number of microorganisms. You know, there are microorganisms in the air, in the water, in the feed, over the animal body, within the udder. So when we talk about the hair follicle itself, into the deep, so we have microorganisms. This entire system, we call it as ecosystem of microorganisms when it comes to the milk production system. And the milk, where the milk is produced, okay, it is produced obviously from the milk animal. Milk cannot be produced by the machines. Okay, so when it comes to a live system, there are microorganisms in the cow urinary tract, cow gastrointestinal tract, the milk animal skin. So all these are the ecosystems. In addition to this, where the animal lives and how many animals they live together, what kind of management practices are followed. So all of them, they are the ecosystems for these microorganisms. That's why large number of the commensal organisms, the opportunistic organisms, And the pathogenic organisms, they all influence the total contingent microflora in the milk. Then many microorganisms, they live a symbiotic relationship with a host animal or the host animal environment. So there are large number of nutrients that are available, and these are microorganisms. They survive there in a climax way. Okay, what is the ecological climax? We'll study a little later. So that there is an ecological climax that has been reached, and whenever that ecological climax is breached, so there is infection of an individual animal or infection of a bunch of animals within the herd. And these microorganisms, when we say they are living with symbiotic relationship with a cow, they are indispensable to these ruminants. You know that how these low quality carbohydrates are they are used up by these ruminant species. It's all because of the microorganisms, and certain vital requirements of the body are synthesized by these microorganisms, so they are beneficial, and they are the one, they modulate the innate and adaptive immune system of these host, okay, that is milk animal. So also in the human being, if probably you make my body sterile, probably I cannot survive. Okay, it's not possible to survive. So there was of late there was a concept of the gut microflora transplantation to have healthy lives. Okay, so in that aspect, if you see, if you talk about the milk and milk products today, we have a large number of fermented products. It starts from idli and goes up to the cheese, the curd, the yogurt, all those things. What are these? These are either microorganisms or the products of microorganisms, which are very much beneficial for the building of the health. So this is what the relationship the microorganisms have evolved over a period of centuries with the host. And when they are present in the milk, there are two things. One is they may not constitute any public health hazard because there is no disease causation in the humans. The second, they may bring about the deleterious changes in the product that we call it as a spoilage, loss in the organoleptic perceptions at the beginning, and later on followed by the marked physical and chemical changes in the milk, making milk or milk product, making the product unfit for human consumption. So with that perspective, you have to see this microbiome. And if you see this microbiome, obviously you can anticipate the type of the changes that you expect in the milk or the milk product. So our traditional thinking, it was just like if at all any infectious agent is present in the cow udder, in the cow or cow udder, we see it as a host pathogen interaction. Okay, we see it as with the perspective of the pathogen itself, but not all microorganisms are pathogens. You know that if you take the total microorganisms as 100% on the planet Earth, more than 99% are useful microorganisms. Only up to 1% of microorganisms are harmful pathogens, and it is these harmful pathogens that bring about the disease in the humans or the animals themselves. So our traditional thinking has been always, when we say microorganism, oh, that's harmful, that perception we have to change. Okay. Next, intramammary infections. That is a disease process that starts. That's why throughout my another 4 or 5 slides I have shown this arrow. As long as the microorganism is there within the animal body, the environment is very hostile. So microorganism, it is very difficult unless it has very strong mechanisms such as Mycobacterium tuberculosis, Brucella organisms. So likewise, there are some evolved mechanisms. It is very difficult to survive within the animal body or animal body compartments. But when it comes to the udder, so when you know that there is the blood mammary gland barrier. So that barrier is preventive, except some microorganisms. So it is preventive. Okay, not readily these organisms are passed into the milk. Then what is the most important is when we talk about the microorganisms, majority of the microorganisms, they go through this route only. Majority from outside to the teat orifice, to the teat canal cistern, then the gland system. So then they will go and get deposited within the glandular tissue. So now the thing, what we got through this translational research in the human, they have found that those women who used to have very healthy microbiota in the gut, they used to have the healthy microbiota in the breast. So leading to the passage of the better microorganisms to the children. So the children used to live healthy. Okay, you know that bifidobacter is one such bifidobacterium, and bifidobacter as a immunomodulant. That is the relationship. This is what is some unknown pathway that yet need to be elucidated. So there is a link between the mammary gland and the enteric pathway. So entero-mammary pathway we said as such systems have not yet been fully understood in the cattle. So until then, we have to see the microorganisms as the extraneous introduction into the gland than endogenous. Endogenous is also possible. The percentage wise, if you talk about it, it overweighs the internal one. Then one more important aspect associated with the microflora of milk and milk products. This is where this public health is different from microbiology and LPT. I am not here to teach you the starter culture and other things. That is the production point. The livestock product technology people will talk about that. And when it comes to the classical isolation identification and all, the microbiology people will deal with that. In public health, I am worried about two things. One is the animal health, second is the human health, and the link between animal and human. So the recent advent of these recent molecular techniques, most importantly the metagenomic perspectives. So the bearing traditional identification of these pathogens that is solely on isolation identification, either biochemical conformation or characteristic colonies on based on biochemical reactions. So that was the way to identify these microbial pathogens. But there are many organisms that could be involved in the pathogenesis, but they cannot be cultivated because of the so many constraints in the traditional microbiological techniques. So recent metagenomic approaches, they have helped us to change our attitude towards seeing these pathogens or the organisms that are living with in symbiotic relationship with the host as host pathogen interactions. So it is not host pathogen interactions. Okay, it is rather the type of complex network of relationships that the organism has with the host. With that perspective, you see, so we have to change our perception towards seeing the contingent microflora of the milk. Then some more discussion on the commensal mammary microbiota. So the ecological properties, they do have their own implications on the pathogenesis of the mastitis that I explained. Then several biotic and abiotic factors that influence the udder microbiota. Okay, we know that what are the biotic factors related to the host, abiotic factors, the presence of nutrients. Okay, and the microbiota of teat canal and mammary secretions, they are all in the form of a niche. Okay, microbial niche, and they will be in the dynamic position. Dynamic position in the sense they will be constantly evolving. So the type of microorganisms that you expect in the first parity will not be the same when the animal is approaching somewhere 8th or 10th lactation. It will be a bit different. Okay, this is what we call it as the dynamic microbial ecosystems. If you change the management, the microorganism ecosystem get changed. If how about these things in India? If you talk about the developed countries where the animal by the time it comes to the production until it is culled, so invariably it will be with a single owner, invariably, or there may be one or two, maximum two changes in the ownership. But in our country, in its lifetime, a cow is sold so many times. In its first parity, it is sold. When it was calf, it was sold. Then second parity it will be sold. Third parity it will be sold. Within single parity itself, it gets sold several times. When it is sold several times and then it visits several ecosystems, so obviously the microbial ecosystem should be very diverse. Okay, it will be A plus B plus C plus D. It is a sum of all those ecosystems. So we can expect much more diversity of microbial flora in Indian situations compared to the Western situations, because the ecosystems are different, the managements are different. Okay, it is true with, okay, the type of microorganism that I harbor in my gut is different from madam's gut, is different from your gut. Within same family, siblings are different because you have a different kind of food habits. The other siblings, they have different kinds. You are staying in a hostel, they are staying in some other hostel. The type of the food is different, and all those. When all these things are different, obviously, this is what I mean. The microbial ecosystems are diverse and it is dynamic. Okay, that's why whenever you are suffering from diarrhea, a intelligent doctor, a highly intelligent and highly legal doctor will never prescribe you antibiotic. Rather, they will give you a lactobacilli or some other probiotic, so that instead of getting cured of diarrhea in 1 day or 2 days, you will take 1 week to get cured. But in your perception, that doctor is a bad doctor because he has cured. It took 1 week time, 15 days time to cure to that diary. Just to give an example, my daughter somewhere around, she was two years old. So she was suffering from diarrhea, and I went to a family doctor, a neighbor of mine. Then he did not give antibiotics. Okay, I insisted a lot. Okay, probably I was wrong in insisting. So he told, no, doctor, it's not at all required. So he gave the probiotic. It took 1 week time, but she got cured. So that's how the things will move when it comes to the dynamism of microbial ecosystems when we talk about. Then earlier I have prejudiced your mind with something known as host parasite interactions. When we talk about the host parasite interactions, the physiological and anatomical parameters, genetical parameters, all of them, they have their own managemental parameters. They have their own impact on the total contingent microbiota. I think it is sufficient, this microbiome concept. So we move to the further concepts. Then, more is the diversity, so more is the contingent microflora in the milk product, milk and milk product. Then, whenever a cow suffers from mastitis, so this is where the relative predominance we talk about. We know by this time the Darwinism, struggle for existence, survival of the fittest. Survival of the fittest means who is surviving here in a mastitis, intramural infection? The one which is pathogen is making use of all opportunities, and it is growing and it is outgrowing in number. Then there are only handful of microorganisms that are present in the entire udder. So that is what is the diversity is lost. So this is what happens when an invasive species enters into, say for example, in a Nile, Nile River, they introduced a perch. Whenever they introduced a perch, all the local small fish were killed because it is a very voracious predator. Likewise, in Karnataka and Andhra Pradesh, they have banned this catfish. Why they have banned this catfish? It is detrimental to the agriculture and the fisheries. Why it is detrimental? This catfish is a non-vegetarian. It is going to eat anything. Even if you are going to throw the dead cows, it's going to eat. Starting from human, animal, dog, any scavenging, everything it's going to eat and it's going to gain the weight. So it also eats the local flora, local fauna. What is this local fauna? Local fauna, diversified fish. When all these diversified fish that play an important role in the balancing of the ecosystem, when they are killed, so there is loss in the biodiversity. So keeping the impact of this catfish on the biodiversity, government has banned that. Okay, these are the invasive species. So likewise, you come to the udder. You just think that Staphylococcus aureus, a particular pathotype, is growing in very large number. So remaining all lactobacilli and all they were living in symbiotic relationship, their number gets reduced. So in the intramammary infections, so invariably less number of pathogens they predominate, and all that diversified ecosystem is lost. Okay, and this is what we call it as a dysbiosis. Okay, dysbiosis. Many times there are some people bad halitosis, a bad odor coming out of the mouth. Why that happens? Invariably, that happens whenever you are on an antibiotic therapy or something like that. You have got you took that. So very few organisms producing certain volatile substances that emanates very bad smell. So like this is what we call the oral dysbiosis. Likewise, dysbiosis means very few organisms growing in number affecting the native flora, that we call it as a dysbiosis. This is a disruption. Bio, life, cis. Okay, dysbiosis, just like symbiosis, it is opposite to that. So during mammary infection, so there is dysbiosis. Few organisms have outgrown and good, and these are bad ones. These are pathogens invasive, and good microflora has been wiped out. Why it has been wiped out? Because of the predominance of these ones. There are many more pathways why they predominate. That is not the discussion today, and for undergraduate level it is sufficient. Okay, then there are several immunomodulatory components in the milk. So we call them as lysozyme, a non-specific inhibitor of the cell wall. The lactoferrin. So iron is required for microorganisms. So this lactoferrin, so it depletes the iron, so microorganisms cannot multiply. Then antimicrobial peptides, small peptides, few amino acid moieties mixed together. Okay, the dipeptide bond, they have the antimicrobial property. We call them as antimicrobial peptides. These antimicrobial peptides are secreted in the milk, so they are also antimicrobial. Then certain oligosaccharides that are there in the milk, so they all inhibit the growth of the microorganisms. So whenever there is a mastitis, there is evasion of all these things. Okay, and even why the shelf life of milk we say 3 to 5 hours at room temperature. This is based on these antimicrobial properties. Okay, when the milk has been obtained from a healthy cow. Okay, so these defense mechanisms, immunomodulatory mechanisms, have been overcome. That's why there is an intramammary infection, and whenever there is an intramammary infection, the number of organisms of few types has increased in very large number in the milk. Okay, now let us understand these structures of. I told you in the beginning, rather than from the cow side, the intramammary introduction of this microbiota is invariably from outside through the teat canal, teat orifice first, teat canal. Okay, let us understand what is this teat canal is. Okay, the teat canal has a very complex biochemical and structural characteristics. Okay, and it is the first line of defense. So it is closed when the animal was pregnant. Okay, that's what in a dry cow therapy we see that it is sealed properly. If it is not sealed properly, there can be introduction of the organisms into the udder. So it is a physical barrier for the entrance of the microorganisms into the glandular tissue. However, there are other factors also. Say for example, you just see in earlier days what used to happen. The people used to lift the water from the wells, okay, open wells with the construction, pakka open wells. So they used to get it through earthen pot. Okay, just imagine some 70-80 years back. If you have seen some villages and all, or if you have visited some historical monument and all, wherever such open wells are present, okay, you can see. Even though the pot was made up of the earthen, a big stone there, okay, had usually if you have seen clearly, you can appreciate. Since I have seen, I am imagining that. Okay, so wherever that the pot used to be, that used to be kept, no. So at that place there is a depression that is created. How come a earthen pot creating such a hole there on the very hard stone? Okay, it is because of frequent use. Several people coming and using the same spot again and again and again and again. So the earthen pot has carved, engraved, okay, has created its own impression there on the hard rock meals. Just imagine a cow giving 25 liters of milk a day, okay, and in its lactation of 305 days, about say for example 6 to 8 or 6 to 8,700 liters milk, and all milk coming through 4 quarters, okay, and these 4 small holes, okay, this is what the teat canal is. When that is the amount with which the milk comes out, okay, amount of milk that passes through, if you use a hose pipe in your garden, within a year that is damaged. Even in front of college also we are using the hose pipe, okay, hose pipe itself get damaged because of the pressure, and every day you are pulling the teat, okay, or you are using the milking machine to get this, and all this milk is coming through this one only. So how robust the structure of this teat canal will be, okay, and any damage and injury to this one, what would be there? This is the gateway. Any damage here would lead to introduction of the microorganisms into the udder, and when these are pathogenic, it would lead to intramammary infection. If there is intramammary infection, the microflora of the milk gets changed. Okay, it's going to increase. That is one way. And second way, soon after milking, there is a mechanism that it gets closed. One thing is that when during dry period, okay, when cow was pregnant, about to parturiate, so 60 days, the dry period, at that time it was sealed, okay, naturally with antimicrobial substances, it got sealed with a plug, okay, teat plug, we can say it. Okay, that was one way. Second way is after every milking, this gate has to be closed. Otherwise, if you keep the gate open, anybody can enter there. Okay, even in your house also, if you keep your gate open, the dogs, the pigs, the stray birds, rat, bandicoot, everything will enter into your house. That's why we have a gate system, no? Like that, there is a gate system. The interior of this one, we call it as the Rosetta Furstenberg. So what happens, soon after milking, for some time, this teat canal orifice is open. So this is a period, very risk period, the organisms enter there. This is why, as a management point of view, we use post-milk teat dipping. So our aim is to seal this because it is open for some time. Some farmers, they have a practice that feeding soon after milking, so that animal is busy and won't sit. If the animal sits, so it is going to sit in the urine and the feces, so organisms will enter from the urinary system and the gastrointestinal system. Is it clear? The teat canal, number one, the physical barrier. Number two, the functionality that it does, opening, closing, that's what is. Okay, and it is the gateway to the udder. So with all these things, we have to see that teat canal is an important, the gate that is important in determining the microbiota or microflora of the udder. Come to the teat apex. Okay, the teat apex means the tip of the teat. So large number of microorganisms have been isolated from this teat apex. Okay, so they have been identified by both culture dependent systems and culture independent. Culture independent means identification of the genetic material, not cultivating the organism on a selective medium. Okay, so 4 important of all these microorganisms, so the bacteria, virus, fungi, rickettsia, chlamydia, mycoplasma, prions, so all that, when you protozoa, when you compare with all of them, the bacteria are far most important. Okay, and that too, bacteria belonging to these taxonomically 4 phyla. Okay, actinobacteria, bacteroidetes, the firmicutes, and proteobacteria. So these 4 phyla of the bacteria have been identified in larger episodes frequency than others. Okay, the common microflora that have been isolated from the Teat effects are the Acinetobacter, Aerococcus, Corynebacterium, Enterobacter, this Phaclamia, Lactobacillus, Lactococcus, Micrococcus, Propionibacterium, the Staphylococcus, and Streptococcus. Most importantly, when we talk about the Staphylococcus, the non-aureus Staphylococcus. Earlier, we used to call them as coagulase negative Staphylococcus. Okay, now the new nomenclature for them is the non-aureus Staphylococcus. Non-aureus in the sense, you know that the genus Staphylococcus. Staphylococcus aureus is the major one. Some non-aureus staphylococcus means they are all staphylococci, but the species is not aureus. That's what it is. Okay. So Staphylococcus chromogenous, Staphylococcus simulans, Staphylococcus xylosus, Staphylococcus hemolyticus, Staphylococcus epidermidis. These are all the important NAS. They are associated with the Teat apex. They are there already, and given the opportunity, given the chance, they will enter into the udder. And when they enter into the udder, they will lead to the mammary infection. When they lead to the mammary infection, their number increases in large millions and millions, and they will be there in the milk. In that perspective, if you keep the udder health, if you keep the cow health, okay, so obviously you can reduce the chances of the clinical or subclinical infections, so leading to lesser number of these microorganisms. You cannot make the milk zero. You can reduce the number. That's important. Next, the teat canal microbiota. Teat canal, I have shown you the anatomical structure here. This is the teat canal, short one. It is teat sister. Okay. It is a gland sister, mammary gland sister, and these are all the alveoli. Okay, many alveoli. It is a diagrammatic representation. You just remember your anatomy, you'll understand that. Okay, so the alveoli, so they will all secrete to the this lumen, so that gets accumulated in this system. Okay, the gland is around say for example 4 to 6 liters capacity, but how come the cow milk 25 liters? Okay, there is a constant secretion of the milk. Okay. Now, when it comes to the teat canal microbiota, it is not significantly different from the teat apex or the teat canal. Okay, it is just like that one only. Earlier also we discussed about this. Again, I am discussing this, and the relationship between the mammary gland and the gut I talked earlier. However, with respect to the milch animals, much more research is needed in this aspect to establish conclusively the gut flora and the microbial flora of the udder. Then, one take-home message from this research is that, okay, you read the articles, you'll understand the latest science. So if you use a lot of probiotics, so you can have the better babies, okay, especially the mothers. Okay, that message, that take-home message, you can get it through this research. Then you come to the milk and colostrum microbiota. So as such, colostrum is not our discussion because colostrum is not a market milk. But even then, it is important to discuss because there are some colostrum-based milk products out there. Okay, colostrum won't enter the normal milk supply chain, but colostrum-based milk products are in the market because it is separately collected, and that too, welfare has to be ensured. Welfare in the sense, colostrum means the quantum. Okay, whatever it is, 1/10 of body weight or whatever it is, the thumb rule, it is there in the management side. That much has to be given to the offspring. Remaining can be harvested. That's what is the practice. Okay, welfare point of view, humanity point of view. So that goes into the market as colostrum-based products. So that's why this discussion is imperative. Now, so when it comes to the contingent microflora of the milk, as I told you earlier, it is A plus B plus C plus, it is summation. What are those there? What is A, what is B, what is C? This already I have discussed. Again, these four major phyla of bacteria, they are important. So in the milk, you expect, and this is the milk of a healthy quarter. Okay, if the quarter is infected with intramammary infection, then there will be few numbers or few genera or few species of the bacteria, but their number will be high. Okay, are you getting me? Their number will be high compared to the routine numbers that is there in the healthy quarters. So routinely we get Staphylococcus, Ruminococcaceae members, the Lachnospiraceae members, Propionibacterium, Stenotrophomonas, Corynebacterium, Pseudomonas, Streptococcus, Commomonas. Okay, the organism is comma shaped. Bacteroides, Enterococcus, Lactobacillus, Fusobacterium. These are all the organisms. It is again a list is incomplete. Major microbiota detected across the globe. Okay. There are some miscellaneous also, which are seen in one or other country. It is global as a whole. Okay. This is from a very good review paper. This information what I compiled. So it is on global scale. Then from the colostrum, Staphylococcus, Provotella, Ruminococcaceae members. Okay, Ruminococcaceae is not a, it is a family. Bacteroideals, okay, it is also a group. Clostridials is again a group. Pseudomonas is a genus. Okay. So in culture negative mastitis quarters, it is healthy. Okay, healthy milk, healthy colostrum. Now you come to the mastitis quarter. Mastitis, animal is suffering from mastitis, but when you cultivate the organisms in the lab, microbiology lab says that it is negative. Then, using the genomic approaches, when they identified, they detected Burkholderia, Spingomonas, then this Stenotrophomonas. These were the organisms that were identified, and it is a compiled data from the globe. Then culture negative healthy quarters. Okay, when you cultivated these organisms, they were negative, culture negative. But the same sample, the second sample of half of that sample when which was subjected for genomic analysis. So they found Pseudomonas, Psychrobacter, and Ralstonia. These were the organisms. Probably veterinary students are not familiar with these organisms. Okay, so as I told you earlier, the milk microbiota of clinically affected quarters is less diverse than healthy quarters. That's one inference. Second inference, the microbial load in terms of number, quantity, that is load is higher in the infected quarters than the healthy quarters. This is the generalization with respect to microbiota. Now, two things you learned in. One, relative predominance of the different kinds of microorganisms in the milk. And second, relative number of microorganisms you understood here. Then, major mastitis pathogens, which is already again we discussed in one of the class, Staphylococcus aureus, Streptococcus dysbiosis, Streptococcus uberis, Streptococcus agalactiae, Klebsiella species, E. coli are the major mastitis causing pathogens. There are many more. Around 200 bacterial pathogens are responsible for mastitis. Okay. Then these microorganisms have positive correlation with somatic cell count in the milk. Okay, means microbial count also increased and somatic cell count also increased. Example, Spingobacterium and Streptococcus. Streptococcus you are aware about. This Spingobacterium you are not aware about. You should learn these things. And traditional books, whatever you refer, okay, those books will not have these names because they have been written some 20, 30, 40, 50 years back. So now you have to have the new names of the microorganisms, especially predominantly bacteria. That's why our discussion is more the man of the match here is the bacteria. So I did not discuss much about the virus, fungi, the mycoplasma and other organisms. They are important, but they are less frequently seen than the bacteria. The second aspect we see that is very interesting: microorganisms with negative correlation with somatic cell count. Okay, what is happening here? Nocardia and this penibacillus infections. There is infection of the quarter. Okay, you know that each 4 quarter of the cow is not connected, so there can be intramammary infection in 1, 2, 3, or all 4. That is why you have to speak about the quarter, not as a udder. Okay, udder is the whole, so you have to speak only in terms of quarter. Left for left hand, right for right hand. That's the way to discuss. Okay, so what they found, even though the quarter was infected with these 2 organisms, that is Nocardia and Penibacillus, but the somatic cell count was not high. Okay, it was in the healthy limits only. So this is also an interesting finding that when we talk about the microbiota. So one inference I would like to make here is, so somatic cell count is not the foolproof indicator of the intramammary infection. But this is the proportion. If you see, the proportion of the positive correlation is very high, significantly high compared to this correlation with the negative somatic cell count when it comes to intramammary infection. Okay, that is why, as a thumb rule, okay, not as a truth, universal truth, as a thumb rule, we take that elevated somatic cell count is invariably linked to the mastitis or inflammatory infection in the udder. Then, with respect to microbial dysbiosis, I talked about. Okay, is it strain specific? That is one more research that has come. It is not even the genus or species specific. Even strain specificity they have identified. They identified in Staphylococcus uberis. Okay, that the particular strain of uberis is associated with the intramammary infection, while remaining all uberis are very much immunomodulatory. They bring about better mammary homeostasis. Means there is no rise in the somatic cell counts or other inflammatory cells or inflammatory modulators in the udder or in the milk. So that was one more new finding that has come recently. So strain specificity. That is why when this research is emerging, then our all labs should be equipped to identify the specific strain, not just identifying genus and species. Okay, we should be more equipped in identifying and in deciphering the ongoing pathogenesis or the type of host parasite interaction that occurs in the dairy cows. Next, the mastitis associated dysbiosis has been linked to Enterobacteriaceae family. Okay, there are many members in this. Again, we have some other discussion in some other context little later, and especially the pathogenic strains of E. coli, what we call it as environmental mastitis. Okay, let us conclude the today's class here. See, when it comes to the sources of microorganisms, okay, the mammary microbiome, biota, what we call it as, is ultimately is a result of the microorganisms that come from cow itself, the microorganisms that come from the bedding material, that come from milker or milking machine, the microorganisms that come from the milker's hand, the air, the environment, including the type of the insects that introduce the microorganisms into the milk, and cross-suckling. At some places there is a practice of cross-suckling. The type of microflora that is existent in the calf's mouth. Okay, if it is a particular kind, same dam and same offspring, that's okay, one kind of microbiota. If there is cross-suckling, it is calf 1 plus calf 2. So that's what is the sum that is the contingent microflora is. So with all these potential sources of microorganisms into the milk, when milk is secreted, okay, milk is secreted, So whether the organism is from the cow itself, or has come from the cistern here, or has come from the teat canal here, or has come from the outer one what we discussed. So on an average, again this list is not complete, on an average the milk microbiota invariably contains these four phyla: Firmicutes, Staphylococcus, this Lochanospiraceae, Luminococcaceae, Enterococcus, Clostridiales, this Aerococcus. These are the Firmicutes. Then Proteobacteria, Acinetobacter, Pseudomonas, and Stenotrophomonas. Bacteroidetes, this Provotella, bacteroidales, Flavobacteriaceae, and Sphingobacterium. Then Actinobacteria, Corynebacterium, Bifidobacterium, this Propion bacterium. So these are all the organisms that are there in the milk. Likewise, the different organisms may be same or maybe some additions or deletions. The teat canal microbiota, then teat apex microbiota, all of them, they are introduced into the milk and they will have the impact. There are two kinds of impact. One impact is that they are going to cause the disease, okay, in the humans through three pathways. One is infection, second is intoxication, and third is toxin-infection. So what is the difference between these and what are the organisms responsible? We will discuss in some other class. That is one way. Second way is that they will affect the shelf life of the product. Means milk spoils early. When milk spoils early, we say that the product has lost its shelf life, and it is count related. Higher is the count, so lower is the shelf life. And there are macronutrients in the milk. There is protein, there is carbohydrate, there is fat, and some other nutrients that are present in the milk. When it comes to this kind of a spoilage, is it the bacterial enzyme converting lactose by the enzyme lactase into lactic acid? The acidity, fall in the acidity, this is what we call it as a developed acidity, milk turns sour. Or is it proteolysis leading to alkaline milk pH, alkalinity? Or is it that there is a ropiness that is formed because of secretion of certain mucopolysaccharides in the milk? Ropiness. Okay. Is it a proteolysis? Is it a lipolysis? It is a saccharolysis. What is that change that has been brought up? And what is the end result? The end product, is it a acid? Is it a gas? Is it the ammonia from the amino acids? So what is that? That determines the sensory perception of the spoiled milk or the milk product that is prepared out of that. So these are all the things, the associated factors. Then this is all with respect to the negativity I talked about. One is the disease in the humans. Is it the TB? Is it the brucellosis? Is it the Jones disease? Is it tick-borne encephalitis virus? Is it a foot and mouth disease virus? What is that? Is it a Q fever? What is that? Okay, that's what is one way, pathogen. Second way is the spoilage and shelf life and loss of sensory attributes. The third and the most important is the beneficial effects. Using this particular milk, am I getting a very good quality cheese? Okay, that's what is fermentation technology and beneficial effects. So these are the other aspects associated with the milk microbiome. So this is with this perspective we have